Good morning. My name is Michaela Bowden. I am the Associate Director at the Center for Molecular Oncologic Pathology, which resides within Dana-Farber. And so CMOP is a center which supports collaborative pathology-based research projects that have the potential to result in applications for targeted cancer therapy with a particular focus on prostate cancer. And so today I'm going to talk about automated RNA-Seq sample preparation for archival FFPE tissue specimens. And we're going to talk about prostate cancer tissue specimens. So prostate cancer is one of the most widely prevalent cancer diseases. There are currently about 240,000 new cases of prostate cancer that will be diagnosed in the year 2013, and of which it's projected that approximately 30,000 people will die. So it's the second leading cause of death in men. There's a higher rate of incidence and mortality in African Americans, and though some information is known about this, it's not fully understood why that is the case. The only established risk factor that most people are able to look at is age, where 60% of diagnosis is found in men over 65 years of age. So currently, right now, the standard practice is routine PSA testing for early detection, but obviously the idea would be to find more valuable biomarkers of the disease. So the major challenge we face in prostate cancer is distinguishing between indolent and aggressive types with a tendency to reoccur. And so most research activities that we're involved in are focused on is better understanding this distinction. Okay, so what I'm going to talk about today is a collaboration that we have with Beckman Coulter. We had a goal to optimize the ScriptSeq library preparation method for FFPE samples of varying degradation. We wanted to develop an automated method for this library on the Biomek FXP platform, and then we wanted to utilize that platform to prepare libraries for a particular prostate cancer cohort that we have in-house. And so the big thing about paraffin-embedded tissues is that this is the routine preservation method that is used throughout hospitals, throughout research. Archive collections are stored for decades, and they're an invaluable tool for biomedical research. Large cohorts of samples are required for any sort of meaningful clinical discovery, but extensive RNA fragmentation and cross-linking have been a limiting factor for clinical translation applications, which would lead to biomarker discovery. And so for paraffin-embedded tissues in prostate cancer, typically there are two specimen collection types. There's a fine needle aspirate, and then there's core biopsies. And prostate cancer cannot be diagnosed from frozen sections due to the difficulty of the morphology, so predominantly the archival collections that we have are formalin-fixed paraffin-embedded tissues. And so globally we have these large-scale cohorts where the prostate cancer clinical specimens have been archived, and in-house we have access to all of these samples with clinical annotation, including diagnosis, prognosis, etc., to help us do retrospective studies. Okay, so to the cohort at hand. So the FFPE specimens that we used here, we had 16 cases where we had matched FFPE specimens and fresh frozen specimens. And so in this talk, I'm going to talk about the FFPE specimens. And from these, we were able to isolate tumor and normal cells from the same case. So these tissue sections were prepared for H&E, and they were reviewed by a pathologist to classify, first of all, identify and annotate where the tumor glands and the normal epithelial glands were, and then to classify the disease state with a Gleason score. And so from these, once this was done, we had tissue blocks were cored. So we used core punches of 1.2 millimeter diameter. So we took 3 core punches from each of the regions of interest, And then from those, we automated the extraction of the RNA using a FormaPure kit and used that, quantified and QC'd the RNA for yield and quality respectively, and used 100 nanograms of total RNA as the input for our RNA-Seq library preparation. Okay, so the ScriptSeq workflow. So the kit that we used is called the ScriptSeq Complete Gold Protocol. It's version 2 of their RNA-Seq library preparation. It states that the RNA input level should be between 100 and 500 nanograms. With FFPE tissues, this can always be a challenge because depending on the degradation level of the sample, you may find that it's not always feasible to get very high yields of quality RNA. But the ScriptSeq workflow is as follows. There's 3 basic steps. There's a ribosomal RNA depletion pre-step and purification of the sample, which is followed by a cDNA synthesis and a round purification. And then thirdly, we have the library amplification and purification. And as you can see, it's a relatively straightforward workflow and can typically be handled in a single day. And this is a big advantage of this method relative to quite a number of other methods that exist, and that it can be done in one day, and there's basically 3 steps. In order to ascertain how, so with FFPEs, you can typically get fairly unclean samples because the level of fragmentation is quite high, so we wanted to assess purification strategies. And so we looked at what's recommended with the protocol as miniloop columns, but we also wanted to look at magnetic beads, looking at the ampure beads versus the SPRI select beads to see if we could do better in terms of cleanup. So initially, before automating this method, we wanted to test on the bench where we were with a representative sample from our cohort, just to establish some of the key parameters. And so up in the top left-hand corner, let's see the arrow. So up in this corner, we have the original RNA. So this is the bioanalyzer trace. And as you can see from this trace, so typically what you would see with a bioanalyzer trace is that when the quality of the RNA is good, you have 2 distinct peaks representing 18S and 28S. And these are usually in a ratio of 2:1, which gives you a pretty high RIN number. In our case, most of our samples look like this. So most of our samples are 5 to 10 years in age. And so all the RNA has completely degraded, and you get this much smaller fragmentation peak for the RNA that remains. And then from this, sometimes it's challenging to generate a RIN number, but the RIN numbers range from about 2.4 up to 4. 4 is the best case scenario for some of these samples. And so from previous experience, we have tested library prep methods and left the ribosomal RNA component there. It's a very high percentage of our sequencing data was around ribosomal RNA, so with the next methods that we tested, we wanted to incorporate a ribosomal depletion step. So what's represented here is the RNA that's gone through ribosomal depletion. And then what we did on the back end is do a purified cleanup, and you can see a very clean, distinct peak representing our ribosomally depleted RNA. So we took this through the whole workflow and stuck to the protocol parameters. So we did a PCR, 12 rounds of PCR, and on the back end we compared 3 cleanup methods. So what you can see here is on the left and right side are the molecular markers, and then here's the cDNA library that was prepared. The big thing here to point out is this large anomalous peak. This peak was present when we used the miniloop columns, and it was reduced, but it was reduced dramatically when we used the bead. So the blue represents the miniloop cleanup, which actually looks like it actually added contamination to the sample. And then if you look at the turquoise and green peaks, this is using the bead-based. Not only do you get a higher yield of your library, you also have none of these contaminating peaks present. And so just to zoom in on that, on the ampure beads, you can see here that contaminating peak is completely eliminated, and we have a nice library that's been generated. Okay, so once we were confident with what we were seeing on the bench scale manual prep, we translated this across to the automated system. And so in our first run, we ran in parallel 8 samples on a single lane of the Biomek system, and this is the same sample representative case that I pulled up here. And what you can see here is that this is the same RNA from the beginning, so this is the exact same trace. When the ribosomal depletion is done on the robot, you can see what the library looks like. This is the cDNA synthesis product after cleanup, and then you can see the library that's been generated. And I think the big thing to point out here is that this library's been diluted 1:10, so it's a far superior yields that we're getting in the library when we run it on the automated system. So once we saw this, we went ahead with our cohort, which is our 14 pairs, and ran all of these in parallel on the Biomek. Okay, so just to give you a little idea of what I'm talking about when I talk about the degradation level in these FFPE blocks. As I said previously, we took 3 cores, were punched from all the blocks and from the annotated regions of interest, so either the tumor or the benign areas, and they were extracted by automation method. And what's plotted here is just ranked the yields, and the yield range is anything from 400 nanograms from 3 cores up to 6 micrograms. So it's a huge range we're talking about in terms of the same amount of tissue going into the extraction method and what's actually coming out on the back end. And some of this is due to tissue morphology changes as you core punch into a block, but a lot of it is due to that these blocks are not all centrally stored. They can be stored in various different places. So they've undergone degradation due to humidity, heat, and just general slow degradation of nucleic acids over time. And so what you're seeing here is kind of representative of the type of samples we're trying to establish methods for. And then if you pull out a number of these samples, so we have tumor normal pairs, so pair 12, pair 24, and pair 11. And this is just to give you an idea of what this previous slide, so this is what the RNA concentration looks like, what this translates to in terms of RNA degradation. And on the bottom, you can see that in the case of sample 11 in the tumor, this hump here and the way this trace looks is that there's actually larger fragments of RNA still present in this sample. But then as you look towards 24 and then go towards 12, the amount of actual degradation increases substantially as you go up through these samples. Sample 12 is the worst sample in terms of degradation that we actually ran, and sample 11 was kind of representative of maybe 20% of the samples. So we had more looking like this, but we had a couple that were really low. And so we really wanted to, a big part of what we're going to do going forward is when we've generated this sequencing data, we want to correlate some of the chromosomal regions and some of the genes that are being expressed to degradation level and see if we can come up with a model for determining which samples are feasible for sequencing going forward based off these types of traces. Okay, so all of the samples were brought through the library preparation method and purified to the back end using a bead-based method. And so this is the quantification data from the cDNA libraries generated. And again, you can see due to extensive degradation and a high level of fragmentation, you can see that the yields from our libraries also vary greatly from anything from 50 up to 1800 nanograms. So again, you're seeing that same spread across the cohorts. And so getting into some of the basic QC metrics from these libraries that were generated. So we looked at fragment size across the cohort, and this is actually relatively uniform considering how the yields range dramatically. So we have a fragment average size range of between 210 and 420, and there's one particular sample on the front end here which didn't perform particularly well, but overall there's a fairly uniform fragment size after amplification and cDNA library generation, which which gave us confidence that potentially the sequencing data that we have here is viable and useful. Okay, again, I have these 3 representative samples. So we have cDNA quality. All of the libraries were diluted 1 to 10. In most cases, without dilution, we had very large peaks. So again, the bottom sample here is sample 11, was the sample that was the least degraded of these 3 representative samples. And what you can see from this is that we generated fairly clean libraries with substantial yield. And as you go, again, with the increasing RNA degradation, you can see that the quality libraries there, just the amount of library you're able to generate is diminished. Okay, so for the RNA sequencing, so we had these 16 library pairs were submitted to CCCB, which is the center here for computational biology, which run the sequencing facility as a core facility. And so we submitted these samples in July, I believe, and then the final batch of samples, they were run in batches on the system. So we just go in a queue system, and the final sample batches were delivered to us in mid-August. And so the instrument details, we use a Hi6 2400 system. We selected paired end 50 base pair reads, and then we used basic computational programs to generate, like top hat and cufflinks, to generate some basic stats. In conjunction with what we got from the core, I also work and collaborate with a bioinformaticist, Svetlana Tekmucheva, and what she did was take the raw data and generate basic metrics and align the reads separately, because a big component of the work we want to do is actually establish new algorithms for looking at FFPE tissue sequencing data. So we don't think that the current algorithms that are designed for highly purified, non-degraded RNA are, in essence, ideal for FFPE samples. And what a big role that she has to play in this is looking at designing new algorithms around FFPE. Okay, so this is just a pretty complicated sample list. So this is so some of the things we look for QC, I guess, is probably the best way to look at this. So we submitted all of our samples as de-identified to the sequencing facility. On this chart, we have the date that they were run on the sequencer. We have the flow cell ID for them. We have the adapter IDs that we use. So these were all done in duplexes. So we used 4 different adapters that are 2 different pairs, and these were all paired up randomly. And then in the comment section, I just have marked where we had a series of original samples which didn't have any reps, and then we actually included a couple of the samples. We had 3 technical reps, and some we had 2. So we have sample 64, we have 3 reps for the benign and 3 reps for the tumor, and then for sample 64, we had 2. So this is just to assess. So part of what we want to do here is also assess sample-to-sample variability within a given lane of sequencing, and then sample-to-sample variability across multiple lanes, and then batch-to-batch variability when samples were run on different lanes at different times. Okay, as I mentioned previously, ribosomal RNA, well, we have tested a number of different library prep methods, and initially when we had started out this testing, probably over 1.5 years ago, we had not thought about potentially removing the ribosomal RNA. Actually wanted to see what the ribosomal RNA component was. So one of the first studies we did actually remove ribosomal RNA. But the result of that study was that up to, I think it was between 40 and 60% of our reads were generated were ribosomal in origin, ribosomal or mitochondrial in origin. So obviously going forward from there, we wanted to then assess the same sample types with the ribosomal depletion in place. So this is just a plot to show that the ribo0 step for ribosomal depletion that was run on these samples resulted in less than 0.1% ribosomal RNA still present in the sequencing data. So even though this looks like it varies dramatically, I mean, everything's under 0.1%, so it's 99.9% free of ribosomal RNA. So some of the QC metrics we wanted to look at, as I said, this was a paired end run, and so what you can see here is the R1 represents the beginning direction with reads, and then the R2 represents the back end. And actually, one thing we did notice, there's significantly more reads on the R1 end versus to the R2 end, so we're still looking into this. But overall, we were able to uniquely align 20 to 60% of the reads to the human genome 19. And so when you correlate this with the RNA input degradation level, it actually correlates well. The 20%, the ones which have low unique alignment, correlate with degradation levels, and the ones that are higher with less degradation. Okay, just to hone in on some of the sequence performance metrics. So I just pulled out a representative sample. So in this case, I pulled out sample 6. And on the left-hand side, so initially when Svetlana takes a look at this data, she aligns everything, and then she goes in and looks at what's uniquely aligned. And so on the left-hand side, you can see the difference, the percentage difference between what's totally aligned and what's uniquely aligned. So basically, the majority of what's aligning is actually unique in most cases, irrespective of direction, irrespective of sample type. And we see this across the board. And then on the right-hand side, we also look at the alignment distribution. So we'll look at the ribosomal RNA component, which is virtually non-existent, and the exon region, the intronic region, and intergenic. And so in most cases here, you will see that the intronic region makes up the majority of the unique reads, followed by the exon and then the intergenic. And again, this is fairly consistent across all the samples. We have run samples previously by different sequencing methods and seen a slightly different profile. We've seen slightly more exons than intronic levels. So at the moment, we're looking at how do we compare different library prep methods to see what the differences are in terms of coverage and percentage distribution. Okay, and so I just looked at one representative sample, and this is for the whole entire cohort. So this is looking at 16 tumor normal pairs, so 32 samples. So on average, there was about 75 million reads found. The 50 base pair paired end reads were obtained, and with 95% confidence interval calculated, the mean unique aligned reads are shown in the table here. So for the mapping to HG19, we have about 39%, intergenic 8.5%, intronic 30%, and exonic 15%. And as I said, the ribosomal component is virtually non-existent. So you can see how distinct these features are across the whole cohort, which was a nice spread. Okay, so I had mentioned that we had run technical replicates, tumor benign pairs, and so in this particular sample set on the left-hand side, we have the 3 technical replicates for the tumor sample, and so what this is we just took all the transcript gene information and plotted each of the technical reps against each other and calculated a Pearson correlation coefficient. And so you can see in the case of sample rep 1 and sample rep 2, we have strong concordance at 0.93. Between sample 1 and 3, we have 0.92, and between sample 1 and 2, we have 0.92 as well. So there was relatively good concordance for the technical replicates. In this particular sample case, these 3 were all run on different lanes, and 2 were run on the same batch. 1 was run on a different batch. For the benign pairing, we saw a fairly similar profile. Again, the correlation coefficients were ranging between 0.2 and 0.3. So all of these scatter plots were obviously logged to the 2 scale, and there was excellent correlation between our technical replicates. In order to sort of dig down a little better into what the best normalization strategy is to compare all of these, all the transcripts across the different samples and across the different plates and the different runs, we looked at identifying a panel of housekeeping genes that could be used for global normalization or sample-to-sample normalization. And so we looked at the normal housekeeping genes, 9 housekeeping genes, so ACTB, GAPDH, LDHA, B2M, PGAM1, ALDOA, PGK1, HPRT, GBIN, BIM, PFKP, and G6PD. These are well-characterized exogenously expressed or endogenously expressed genes that should be prevalent in most sample types. And so what we looked at is we looked at within a given case, so the tumor normal pairs, we looked at the correlation of these 9 genes within a case. And so what you can see here is I've plotted 6 representative samples and the correlation of these 9 genes across those samples. And so generally, in most cases, with the exception of 2 sample pairs that we had, this gene normalization gave a linear correlation above an R-squared value of 0.9. So initially, we had thought that using a global normalization strategy initially for this QC would work, but we, based off this and based off the fact that we had suboptimal results for this, we decided to use a normalization strategy for each of the individual samples in and of themselves, and then apply a global normalizer afterwards. Okay, so in order to assess initially what was going on with these samples, we have a panel of hallmarks of prostate cancer, so which includes in and around 100, 110 genes which are associated with prostate cancer progression, diagnosis, lethality, and so initially to assess the quality of the data and initially to assess if there was overarching themes around what's going on between the tumor and benign sample populations, we extracted the information, the FPKM values for this particular gene set. We normalized all of these genes, so I said that 9-marker housekeeping gene panel, and then we normalized. We averaged we got the mean of all the tumor samples, in our case of 16 tumor samples and 16 benign samples, and then looked at the differential expression between the tumor and benign groups as a whole, and plotted. And so what you see plotted here is the log2 differential between tumor divided by benign for this panel of around 120 samples. And what you see here is a threshold marked in, which represents plus minus a 2-fold differential as a threshold. And so what we really wanted to do was just look at, for these prostate cancer hallmarks and ones where there was significant differential expression observed, would we be able to see, was this statistically significant within our cohort? And so the 6 genes that we initially are focused on is EpCAM, Wnt5A, keratin 15, TGF-beta3, P63, and VEGF-A. And so when you go in and look at the tumor and benign populations in and of themselves and generate some statistics and box plots on these, you can see in the 5 out of the 6 cases that we have here, we had statistically significant differential expression. So in the case of the outcome samples, when you compare the benign population within our cohort to the tumor population, we have a p-value that's greater than 0.001. For the keratin 15, actually for the keratin 15 and for the p63, the statistical significance was, extremely high between these two, and then also less significant but still statistically significant was the TGF-beta 3 and VEGF-A. And so where we are with this right now is that we've just completed all the QC on these samples, and the next phase is now to go back to the transcript data that was generated and the isoform data, and actually, with confidence, use these prostate cancer-associated genes as validation to go back and look at maybe novel genes that are differentially expressed that are not included in our hallmark of prostate cancer in our panel, and also to go back and then look at subpopulations within our cohort. So this was just looking at separating tumor benign pairs into two groups. But what we would like to go back in and do, and there's a number of things we want to do. We want to, within our cohort, we have African American and European American subpopulations. We want to go and separate those out and look at the differential expression between the African American cohort and the European American cohort. We also want to go in and take a look at if you stratify relative to the Gleason score, so how, there's low-grade prostate cancer diagnosis or high-grade prostate cancer diagnosis, can we differentiate these populations and look at the genes that are differentially expressed there. And then another aspect of all of this, as I said, was that we wanted to look at all the sequencing data and see if there were methods in which we could interpret, first of all, what's lost relative to our fresh frozen counterparts. So as I had said, we have fresh frozen samples that were also RNA-Seq libraries were prepared and sequenced. So we have that data as sort of a benchmark, and we want to compare this FFPE data to that to see what's being lost through degradation for these patients, for these samples, and then if there's any algorithms that we could sort of predict based off degradation level what you should expect. Is there chromosomal regions that would be affected inversely? Does the size of the gene transcript relate to degradation levels? Is the GC content the issue that we're talking about here? And so on and so forth. So these are all subject to further studies that we are currently working on. Okay, so the follow-up with here is, we have data analysis that's in process, and as I said, we have a plan to compare these to fresh frozen matched cases. We want to look at developing these novel algorithms that would handle FFPE specimens better, and also more importantly, if we can look off the, if we look at the QC of the RNA from these FFPEs, can we make a decision, a go or no-go decision around whether they're feasible for sequencing or not, based off what that degradation profile looks like. And then obviously any sort of validation signatures that are found, we'd have to do a biological significance assessment and follow up with some validation of any gene targets by RT-PCR. Okay, so as I said, my collaborator in all of this is Svetlana Tekucheva. She is our bioinformaticist, and she also works at Dana-Farber. From the Beckman side, Bina Lee was the senior application scientist who helped us get going on all of this. Ashley Rossi is a technician who run all the automated processes in-house. Sarah Simmons came in and helped with the method development and implementation on our Biomek system. Our prostate cancer specimens actually come from the Gelb Center, and the cohort was defined by Matthew Friedman and Mark Pomerantz, who are investigators here at Dana-Farber. Massimo Loda is the director of the center. Okay, so with that, I'm just going to open up the Q&A section and see what questions are coming in. Okay, so I'm looking at a question here. here, which is from Magnolia Bostic, Clonetech Laboratories. So have you ever tried additional DNase treatment of the FFPE RNA prior to library prep? And do you know the difference between your intron and extron is library prep and not sample? What are the other library prep methods you use? Yes, so have you tried DNase treatment of the... We have tried additional DNase treatment of the FFPE samples, and we haven't found, we've had limited success with that, with the samples that we've looked at. Yes, we do know that the difference that we see at the intron and exon level is relative to library prep. I think what we want to disseminate, because we don't have a direct, we don't have samples from both cohorts that were run on different library prep methods, we don't have a direct comparison to make, because we wanted to also understand if it's just something we're seeing in terms of the relative degradation of those samples as well. So the idea we have now is to run a small cohort across multiple different library prep methods to compare directly what's happening in those, but we do know that it's a function of the library prep. One of the library prep methods that we use. So we've looked at the TrueSeq method. We've looked at Nugent, Ovation. We've looked at a couple of different things, and we're starting to look into other novel methods as well. I think a big push for what we're doing is we need to focus on prep methods that will allow us to go to sub 100 nanograms, because a big focus and push of the research we're doing is looking at tumor heterogeneity and looking at tumor microenvironment. And so we want to move to increasingly smaller number of cells and look at the differences between sub-cell populations, maybe within a gland, within a grade scoring. You know, for a given patient, there may be a number of tumor glands, and there may be a number of different scores or Gleason scores applied to each of these glands. So we want to go in and be able to look at different foci within an individual patient. And to do that, we have to be able to extract smaller and smaller amounts of material, and as a consequence of that, have to look at downstream applications where we can have an input level that's much smaller. So the next question I have here is from Marissa Needham from the University of Virginia. And so what method did you use to deplete the ribosomal RNA? And so we used Ribo0. Ribo0 is the method we used to deplete RNA. We've looked at different ways of doing this, but the ribosomal Ribo0 method is by far the easiest, simplest method, and we've had very good results with it. How do you quantify the residual amount of ribosomal RNA in the total RNA prep after purification using the RNA chip bioanalyzer? So we don't quantify the ribosomal. I'm not sure I get the question completely, but if we don't quantify the residual amount of ribosomal RNA, we qualify it. So we just use the Agilent bioanalyzer system, and we just look at what the trace looks like. I think that's the question. I'm not entirely sure if that answered your question, Marissa. I mean, maybe you can write another one if that wasn't clear. Okay, so I think that's all the questions that we had. So I just want to take the opportunity to thank you all for listening, and thanks again to everybody who contributed to this work.